Yeah, the exam. 2 weeks from today. Remember, that's the official time for this lecture section. My other lecture section, its official time is the Wednesday, and I did give you the opportunity to go to the other one if you really feel desperate, not really ready on Monday. I'd rather have you come on Monday and take it, but I'm not going to penalize you if you decide you.
You're going to have a really full class Wednesday.
But you have other classes too, probably, so you may not have the luxury of taking the other one, but I'll give you that. We'll do a study session Friday, the Friday before. Remember, this week and next week are the last week of classes, and then that next week after that is just finals.
So it's next Friday? Bless you.
So next week, Friday of next week.
Okay.
Friday of next week will be the study session. We can go as long as you need, and again, as I keep saying, email me with questions too. I don't mind, and I've already done it. I've done a lot of sending things in the past. That's fine. Okay, so we covered everything that I wanted to cover in this lecture, but I just wanted to review a little bit. I'll say review from last lecture, just because it's probably going to appear on the final, and I also want you to understand what it's about. Okay, and so last lecture we were talking about human evolution. Does anyone recall that?
Yes. And then we went on to
a long time ago. It was last week.
Lamarckian cultural
revolution. And remember there were 3 different populations of closely related species, or maybe subspecies. We're not totally certain. And then the other 2 species sort of died off around 40,000 years ago. Somewhere around 40,000 years ago. And it's about the time when modern humans, Homo sapiens sapiens, came in contact with the other 2 populations. So that looks this isn't part of the Great Leap Forward thing, but it does make it look like modern humans were kind of responsible, or partially responsible, for the extinction of those other 2 subspecies. Well, whatever. It did happen. It might, and we don't know for sure that it happened. It may be that they were just outcompeted. Who knows? We don't know. But it's sort of an awful thought. But it's not an unlikely possibility. Modern humans got ready to cause the other 2 species to become extinct.
It's happened since.
But and modern humans have been on this planet somewhere around 250,000 years, roughly. Okay. But something seems to have changed. Okay, so Homo sapiens, or Homo sapiens sapiens, I'll just say Homo sapiens, has been around, I don't know, that seems sort of a scientific term, has been present on this planet, about 252,000 years. There's not an absolute date. No one has any absolute date on this. So we'll say about this. Okay. There wasn't a whole lot happening in terms of the way they were interacting with our environment. They were making stone tools. They were doing various kinds of things, but there wasn't a lot of change. What we call cultural evolution. That's the next word I was going to talk about. But there doesn't seem. Again, we're talking about looking at the fossil record, looking at when they dig up remains and doing anthropological studies, it doesn't seem like a whole lot was changing, or let's say it's changing very slowly, until somewhere around, somewhere around 40,000 years ago. I think I might have given you the date, 35,000 years ago last time. Again, there's no absolute date, but we'll say some change appears to have happened, but we don't know for sure. It's kind of a guess. It appears to have happened, say about. Again, there's no absolute date here, 40,000 years ago. And after that, there was rapid change in things that these modern humans did. What we call cultural evolution. some change appears to have happened about 40,000 years ago. And after that date, rapid change in cultural evolution. Now remember, when we were talking about evolution before, we were talking about organic evolution, change in gene frequency, et cetera, et cetera, et cetera. But cultural evolution, that's the change in what, how we're living our own lifestyle, and I mean, look at the amount of changes that occurred in your own lifetime. Things like cell phones have become totally ubiquitous in this planet.
Yeah, how did we get along without them?
And how could we get along without them? I mean, this is such a, and if you go, just computers have changed so radically in the last 20, 25, 30 years, and you go back, however long you want to go back, and huge changes have occurred. All that is part of what we call cultural evolution. And remember, what I said about cultural evolution is it changes. Well, you don't have to reinvent the wheel each generation. You take what you have now and improve on it, do more things.
Mm-hmm.
And so that's Lamarckian evolution.
Acquired traits.
That's the evolution of acquired characteristics. Mm-hmm. Remember we talked about with respect to organic evolution, Lamarck's ideas were not correct, but his ideas do seem to be the way things work with respect to cultural evolution.
Mm-hmm.
So, now, first of all, what is this? As I say, some change appears to have happened. Some change. And what one hypothesis is that it's a reasonable hypothesis. We're not sure exactly how it might have happened, but some change may be in the structure of the vocal cords or the vocal box or whatever to allow language, speech, so that you could then communicate with words as opposed to just grunts. I was talking to someone just this weekend. Her son was playing tennis against my son. They're both freshmen in high school and they're 15 years old. And I was saying how God is so hard to get my son to communicate. Tell me something.
He's
and
emo.
She said, yeah, well, she said, my son, I mean, most of his communication skills are grunts. Yeah. And I said, yeah, we get a certain amount of that too. So, well, I know, even with text messages, he just gives me like a little one word, I'll say, give me some details, and there's one little word like K, yeah, but there's no explanation.
Well, there is. It's scientifically proven that at age 13, parents become lame.
That's it. It's parents' fault. I mean, I've lost it. I know that. I realize that. But anyway, no. But here's
in 10 years, yeah.
But think back. Yeah, I mean, you look at things like gorillas. They're physiologically, and anatomically very similar to us. They're stronger on the average than we are, okay, but still, in terms of everything except their vocal structure. Chimpanzees too. They don't have a vocal box or vocal cords that allow this kind of complex speech that we can use.
Mm.
And again, think about the complex speech. It's an abstract thing. You have words that have no intrinsic meaning. They only have meaning because you know what they mean and you know how to use them. But then you go ahead and use them. But what does that allow you to do? It allows you to plan. It allows you to get together. It allows you to provide history. It allows you to explain things, in complicated sorts of
pattern
ways.
recognition.
And so anyway, the point is that if this is what happened, if this change, I'm not saying maybe because we don't know, but maybe in the vocal box, to allow language, communication, but abstract language. But we'll just say to allow extensive communication,
yeah,
more complex communication, far more complex communication.
Yeah.
Actually. Then you can certainly imagine that things are going to be a lot different in terms of just your normal society, the way you're going to interact with the way things are happening. And of course, keep in mind that humans or with in general that we start out as very very immature in terms of our. Some of us stay immature. I have to admit that's true. But anyway, but we start out in a very immature state, and we have to stay with our parents for a long period of time. So there's a lot of learning going on. There's a lot of transmission of knowledge, and with this complex communication, it can be very extensive kind of transmission of knowledge. And you can imagine that somebody comes up with some idea for some new weapon, and someone can after that can improve upon it, and we go on and on and on and do whatever. Could be some new way to fish, some new way to hunt, some new way to raise food, whatever it might be. And it seems to have happened somewhere around 40,000 years ago, such that cultural evolution in the last 40,000 years, in fact, if you want to even just go back to like maybe 18,000 years ago, cultural evolution has changed far more in those last 18 to 20 to 30,000 years than it did for the previous all the previous time that modern humans have been here. So you can say that for 200,000 years there wasn't a whole lot of change in cultural evolution, and then the last say 50,000, if you just want to make it easy, the last 50,000 years there's been huge change in cultural evolution. And of course, as I said, our society just gets more and more and more complex every year, and things change so rapidly. Anyway, yeah, go ahead.
40,000 years ago, there wasn't culture in existence, was there?
Well, I mean, there's still, you can call it culture still. It's different from what you're thinking of
just
now.
A very
so
primitive
complex,
culture.
But still, presumably most of the, there were probably groups, tribes, whatever you want to call it, whatever the word might be, that traveled together. They, stayed together, mostly related. That's the way, if you look at the modern apes, that's the way they are. That there's usually a family group, and sometimes the the young females might go off and mate with someone else. They'll join some other tribe, but so, or maybe sometimes the male might. It depends upon the group. But they tend to stay in.
So wow,
I think with
that's
monkeys they call them troops. I'm not sure if they
do.
Troops
They do.
Chimps and gorillas. But anyway, they tend to stay in. So they certainly have a culture. And what was the thing we were just? I guess it was in the other class. There was something on the web of a bonobo that was putting a marshmallow on a stick and could.
Yep, my mom told me about that.
Oh, the bonobo chimp, yeah.
Yeah, I made a marshmallow and my mom went into all the details. Like they knew it was too hot and they'd blow on it.
Did it cool it off? Now, did he actually make s'mores?
Yeah.
That's what I want to read.
I don't know about making s'mores part, but
did he have some chocolate? Make some s'mores? Did he have graham crackers? Anyway, no, but some of the bonobos and another actually, there are all kinds of examples of the various primates that one primate will learn something. For example, there are these macaques in Japan. And this one female macaque learned that if she took, I think it was rice or whatever, and put it in water, she could wash the sand off of it. And she taught that to her kids, and then that started getting spread around. So now they all do it. So they have culture in that sense. They have
you're right.
But in terms of how rapidly things change, slowly. And even for modern humans, it was presumably fairly slow until suddenly it seemed to go very fast. This is what it appears.
Mm-hmm.
It could be that it just, it's an illusion, but I think it's probably accurate. So cultural evolution, I want you to understand that when we talk about cultural evolution, it changes in a Lamarckian way. And into this, I hope, will make you go back and look at what Lamarck said.
About acquired traits.
Which is the passing on acquired traits. That doesn't happen in organic evolution, but organic evolution is very slow. And I want you to keep remembering that. It's very, very slow. And that's one of the hardest things to understand is how slow it is. There's been very little organic evolution in the last 250,000 years in modern humans. There has been some, and maybe some in the vocal box for that matter. I don't know. But you look back a million years and then you see a lot of changes. Anyway. Okay, so that's I just want to kind of review that. I know we talked about it, but I just wanted you something I want you to think about. Yeah. I was just going to go do that, actually. Yeah, I was just thinking the same thing. See how great minds just, yeah.
Think like.
Okay, so let's, what I want to do now, in the next several lectures, well, 4 lectures, right?
Yep.
2 this week, 2 next week,
Yep.
is talk about the diversity of life. That's going to be the topic, and we'll cover, very briefly, various different organisms. Today, a little bit on viruses, and then a little bit on bacteria. And then next time we'll talk a little bit about archaea, and I think we'll talk a little bit about protists. We'll talk a little bit about plants, and a little bit about animals. Obviously, just a tiny bit about everything, because any of these ideas is you could spend a lifetime studying viruses, for example, or whatever, depending upon your interests, of course.
Bless you.
I mentioned in my other class that when we were talking about them in lab, that I like viruses, and I don't mean I like to have the virus. No, I don't. In fact, the only way we know about viruses is because they cause diseases. So they're not nice things in that sense. But I like them because they're fascinating to study. And it's very possible that viruses have had a very big effect on evolution, too.
It would seem as they did for thinning the gene pool.
Quantify, but they probably had a big effect on evolution. But I thought I would just tell you a little bit about viruses, a little bit about their structure, a little bit about how they work. So I put this over here, just to say something about that. Then we'll talk about bacteria. And again, in my micro class, I would teach, I would do about 3 weeks worth of lectures on viruses and about 3 weeks of lectures on bacteria. Because these are huge topics. But anyway, let's just talk about viruses. So viruses, these are not included in the normal classification scheme. Why not? Why are they not in the classification scheme? Why?
Because they're not actual cells.
They're not cellular, right? So they're not cellular. So therefore, they're not in the normal classification scheme.
The nucleotides and
said the very first lecture, I think,
coatings.
we said that the cell was the basic unit of life.
Mm-hmm.
So it's always been the problem. How do we classify the viruses? They're obviously biological entities.
Yeah.
And they're made of biological molecules, but they're not cells, so we can't include them. Actually, when I teach micro, I do make a classification scheme in which you can include elements that are not cellular, but this has been proposed by somebody else. I didn't come up with the idea, but so you have a separate category. Elements that are not cellular but do have some control over their own replication. Well, we'll see that that's what viruses are. So they're not cellular, so they're not usually included in the normal scheme of taxonomy or classification. They're called obligate, this is a term for them, in the old days, and we still use the word, obligate intracellular parasites. By the way, there's some bacteria that fit this category too. So just because something is an obligate intracellular parasite doesn't guarantee that it's a virus. But what does this mean? And this word intra, I think I mentioned this the other day. Intra means within.
Yeah. In the cell.
As opposed to inter.
Inter.
If you see intercellular, it means around the cell. This is intracellular, so it means inside the cell. Obligate means it has to. Has to what? Has to be inside a cell. And when it's in there, it's a bad guy. It's a parasite, not a good guy. Bad guy. So a virus. So viruses are completely inert outside cells. There are viruses all around. They're doing nothing when they're somewhere outside a cell. They're just sitting there waiting for their big moment. And their big moment is when they find the proper cell to get into. Okay. How do they know what cell to get into? What is the proper cell? It's say it again louder. It's the R word in my physiology class. Receptors. They've got to have the right receptor, and their cell has to have the right receptor so that they can do it. No, I mean whatever. So viruses are inert, meaning they don't do anything outside cells. So they don't do anything outside cells.
Do they degrade outside?
Well, they can degrade, yes.
See, that's the thing that
And-
if they can die, that
Well,
kind of does make
But
them.
when we use the word live virus versus killed virus, or what that means is a live virus means it can, if it gets in contact with the right cell, it can cause an infection,
replicate.
whereas a killed virus cannot. So we're not using the words live and dead in the same sense as we would for a cell.
Okay.
But even with a cell, a live cell means it can go ahead and do its normal functions and can reproduce and all that. Anyway, so viruses are inert inside cells. But I'll say once inside the correct cell for whatever virus we're talking about, and most viruses, not all, but most viruses are very highly specific with respect to what kind of cell they'll infect. Usually if it's a multicellular organism we're talking about, they're usually very particular about what organism they'll infect, and within that organism, which cells. Anyway, but once inside the correct cell, the virus takes over the cell and turns the cell into a virus factory. I'll say turns the cell into a virus-making factory. And notice it turns the cell into a virus-making factory. The virus doesn't do any work at all. The virus only provides the information. What is the usual form of information in biology? Say it again. Well, nucleic acids will be general nucleic acids. Information in the form of nucleic acids. So the cell does all the work. The virus just provides the information. I just said info, but information. In what form? In the form of nucleic acids.
Has anybody ever found a beneficial virus?
Depends on whose perspective you're talking about the word beneficial. I'll give you an example of one that you would say is beneficial. Just one second. In the form of nucleic acid. So here's an example of an interesting virus. They only discovered this maybe, 15 years ago or maybe 20 years ago, so it's fairly recent. But so here's something that they've known for hundreds of years, what are called the ichneumon wasps. You may have heard of these, and there's some other examples that are similar to this, but the ichneumon wasps are the ones that will find a caterpillar, and then they'll sting it, paralyze it, and then they'll inject all the eggs inside the abdomen of this caterpillar. And here's this caterpillar still alive. And when the eggs hatch and the larvae eat out the insides of the. I know it's revolting to think about. They eat out the insides, and then they eventually come out.
Also,
and then
the
they turn into
tarantula.
wasps, and they go do the same thing. And this really bothered Darwin. And he said, how could it? He said he could understand how these things could occur through natural selection, just some random kind of things that happened over millions of years, But he says, I cannot imagine a creator being so, what he would call it, mean or whatever, to this poor little caterpillar. Yeah. Not that we usually have too much feeling for a caterpillar, but the very idea is just revolting. Yeah. But anyway, whatever. But what they just found out about 20 years ago or so is that there's actually a virus involved, and when an ichneumon virus, it's a insect virus. There are all kinds of insect viruses that they know about, and they're billions more they don't know about probably. But anyway, so when the wasp carries this virus, it doesn't do anything to the wasp, but when it stings a caterpillar, it's actually the virus that causes the caterpillar to become, paralyzed. And then when the little larvae eat the cells, they get that same virus back again, so that they can pass it on to another, caterpillar so they can.
Wow, so they actually have to take that in, they don't have it, or that's crazy.
So in this case, that virus doesn't hurt the wasp at all, but it does have a big bad problem for the caterpillar. And that's why I said depends upon whose perspective. So from the perspective of the wasp, that's a good virus, right?
Yeah.
But from the perspective of the caterpillar, bad. So, as I said, it's just weird that it turned out to be a virus that's involved in this. It's just, that no one expected that. When they found that out, they were like, whoa. Anyway, but back to viruses. So viruses don't do anything by themselves. All they do is provide information, and the information is nucleic acid, the usual kind of information. So what is the structure of viruses? Let's talk about the structure real quickly. They're made of there's a protein coat, and it's called the capsid. Surrounding the nucleic acid. Now, here's one little weird thing about viruses, and I've said this, I don't know if I said it in this class, but I think I did, that if you ever get any rules, so to speak, about how things work in molecular biology, if there are ever going to be exceptions, there are going to be exceptions, or almost for sure there are going to be viruses, and there are all kinds of exceptions in viruses. So for example, surrounding the nucleic acid is going to be RNA or DNA, but not both. Okay. And that's different from, our cells. We have both RNA and DNA, right? So living organisms are made of both RNA and DNA. The DNA is usually the information, right? The chromosomal type of material. But we use RNA to do all the protein synthesis. But there are viruses that are what we call RNA viruses. There are viruses that are DNA viruses, but we never have both. Now, the protein coat, the capsid. It looks like this is a typical picture that we'll draw. Make it simple. That would be the capsid. And here's the nucleic acid here. Now, this is a two-dimensional little drawing. What do the capsids usually look like? There are two main structures. They're not the only two, but these are the main ones. The vast majority of viruses fall into one of these two categories. So the capsid, I'll say two main forms of the capsid. One is icosahedral. Oh, now there's a good one for you. Icosahedral, or helical. Now, what's icosahedral? What's an icosahedron? Oh my God! I used to teach geometry, just so you know.
I was going to say it's a shape.
There were four. I never played Dungeons and Dragons, but if you played Dungeons and Dragons, did anyone play that game ever? I never have. Okay, and the little pieces that you use, one of them is an icosahedron, I think. They're what they call the 5 platonic solids.
How many sides does it have?
20.
Yeah, that's the 20-sided die. Yeah.
Yeah. So it's 20 sides, but it's more or less. It's more or less shaped like a soccer ball. Roughly.
Yeah.
Okay.
Yeah.
And this is the easiest way to describe it. Or you can think of it as a spherical. It's less than spherical. It's more like it's a little less than a sphere. It's more like a little soccer ball, which is pretty spherical. Actually, soccer balls are made of. But it's still warm. Why don't you open that door? Open that door. See, there's the thing I think to keep it open. It is warm in here. I agree. She's getting it, Amy, she's getting it. It is warm here, actually. We'll finally have a nice day.
Right.
Anyway, but just for interest sake, nothing you guys need to know about, but there are what they call the 5 Platonic solids. These are the perfect pure solids. One of them, and one of them is a cube. It's made of 6 squares, perfect squares, right? That's what cube. You all know what a cube looks like. Another one is a tetrahedron. It's 4 equilateral triangles. And then there's the
4-sided die.
Dodecahedron, which is 12 pentagons, 3. Then there's the octahedron, which is 8. No, but octagon is 2. It's just two. That's only 2-dimensional. This is 3-dimensional. So it's an octahedron, and that's 8 equilateral triangles, if I remember correctly. And then there's the icosahedron, which is 20 equilateral triangles. Anyway, you don't have to worry about this. I'm just spouting off this stuff because I think it's cool stuff.
Well, yeah.
Anyway, whatever. So you have these
stack
two
the
main
2
shapes.
4-sided dies on top of each other. Get 8-sided. Get 20.
And the helix. So, if it's a helical one, then the protein kind of surrounds the DNA, kind of like in a helix. I'm not going to draw it. That's not a big deal for my purposes. One other little bit, and this actually becomes sort of interesting if I talk about. Did I say anything about? Well, I didn't. I said bacteria in humans. I was going to say I could have said viruses in humans, but I'll say this. If you just have a capsid, if you just have the capsid surrounding a nucleic acid, that virus is said to be, oh God, here it goes, be ready, this is pretty embarrassing, naked. It's a naked virus.
Naked virus.
Oh God. So, but some viruses have it's like an envelope. It's called an envelope, but it's like it's a membranous structure. It's not officially a cell membrane, but it's a membranous structure. So I'll put this over here. We're still talking viruses, so I'll leave the word up here. Okay.
Got all this.
So we'll just say some viruses have a membranous, membrane. I'll say like a membrane covering. I'll put that one. Membrane.
Is that the protein?
It's phospholipids and proteins covering called an envelope. Now it's not a real big deal here for our purposes, but the point is that I want to say a little bit about it. So let's first, anyway, there said to be envelope. So we either have naked viruses. This would be a naked virus. If you have an envelope around it, I'll do that like this. This would be an envelope. And I'll put in parentheses, some viruses have this. Not all. So what's the deal? I said that viruses get inside the cell and they take over the cell and convert the cell into a factory. In some cases, what they do is, the cell will just make all kinds of new viruses. Then the cell bursts. So the cell doesn't like this; it kills the cell, but it releases all these new viruses. Those are naked viruses. Some viruses, they don't kill the cell; they make new viruses, and the viruses bud out of the cell. You don't need to know this for the test. I'm just going to show you real quickly. Show you where the envelope comes from. So if we say this is let's say I did it blue. So let's say this is the host cell. And so here is our virus here, and you have all these new little viruses in here. They can be a lot smaller than this, but I'm just trying to show you these are the viruses. And they'll move up to the edge of the cell here, and they'll kind of push their way out. And as they push their way out, they'll go like this. And then the next picture would be just they've escaped, and we're out of the You have this, and then you have this like this. It
And they're suddenly inside of a membrane.
exocytosis. You remember that exocytosis
Yeah.
thing? They've actually pushed their way out, but when they push their way out, they picked up a piece of that cell membrane. Now, it's not just cell membrane because there are in it various kinds of proteins that are not from the cell that came. I mean, they came from the virus, but they're in that little membrane structure. That's important, and I'm going to tell you why, just for interest sake. Not you need to know this, but this is just for interest sake. We'll come back to that. So this is how they get their envelope. And I said it's a membrane covering. It's the envelope.
That's how they
They get it by budding out of a cell and taking a piece of cell membrane that's slowly surrounded. But that cell membrane, or that membrane, does have some virus proteins in it. It's got phospholipids and cell proteins, but it also has some virus proteins.
It doesn't have mitochondria. Or
No. No, it's just that little virus.
and the
And as
membrane
it pushed
that
its way
is
out,
stole.
it just covered itself with a
It
little
kind of like steals it.
invisibility cloak. No, I wouldn't call it that.
But I mean that's like the way it's taken in, and when it
Yeah,
leaves, it kind of steals a piece of the membrane with it.
Yeah, it just takes a little piece of membrane. So it's not a cell. It's not a cell, but it does have a little membranous covering. Now, is that a big deal for our purposes? No, it isn't. Except
It's interesting.
in trying to understand certain things. But before I do the understanding things, let me talk about this nucleic acid just for interest sake. I want you to know what the structure is, but I'm not going to worry about all this other detail. I'm telling you a lot of things so that when you read about them, it'll make some sense. But the nucleic acid. Now, I think I told you, and if I didn't, I should have, that in general, this is true, except for some viruses, because remember I said any possible exception would be viruses. DNA is always double-stranded in nature.
Mm-hmm.
RNA is always single-stranded in nature,
Mm-hmm.
except for some viruses. Okay.
Yeah, I remember you
So
saying that.
nucleic acid, you can have single-stranded RNA viruses, double-stranded. I'm going to write with those RNA viruses. So SS stands for single-stranded. And DS stands for double-stranded. So there's some viruses that are their nucleic acid is a single-stranded RNA. Example would be polio. And polio is made of exactly 5 genes. That's 5 genes. But it's an RNA. So it's a single-stranded RNA. Double-stranded RNA viruses are not as common. There are some, none that you need to worry about. I think if you've heard of Brio virus, that's one. There are also some single-stranded DNA viruses. And as I just said, DNA is always double-stranded.
So
except in the case
how
of,
is that distinction made that it's DNA?
oh, it's still made of DNA. It's not RNA. I mean, remember they're different. The DNA has deoxyribose.
Oh, okay.
Versus
Yeah.
ribose, and then you've got U's, and the RNA has U's where you expect to have T's.
Geez, okay. Yeah.
So double-stranded DNA is what you'd expect, but we do have some viruses that only have one strand of DNA. So this is a single-stranded DNA virus. And just to show you how specific things are, and you don't need to worry about this, but when I was on sabbatical at Cambridge in England, I was in a virus lab, and they were studying flu. And flu is a single-stranded RNA virus, and it happens to be what they call the negative strand. Not that that necessarily means anything to you, but the positive strand of RNA is what messenger RNA is, okay, just so you know. And the single strand would be the complement of it. And the guy who was the head of the lab, he's very famous in the field, he was setting up an international conference for the next year or 2 years later that was going to be held in Cambridge. And they hold a lot of international meetings because they're a very famous place. But this was the negative strand conference. In other words, all the people who were going to come to this conference were people from all over the world who are studying viruses, and they're studying RNA viruses, and they're studying only the negative strand type of RNA viruses, not the positive strand. That's how specific the field is. I mean, people in the field are studying just one little aspect of whatever the field is. It's just incredible to me. But there are people all over the world studying this and learning all kinds of things. And many of them have some very important, first of all, you learn a lot about molecular biology studying viruses. You learn all kinds of new things you didn't know before. But then also, viruses. How do you know about viruses? They cause diseases. So, you're learning as much as you can about new viruses to try to fight them. That's basically what you learn. So in general, I actually already said this to you without going into a huge amount of detail. How are new viruses made? The virus gets into the cell. So let's say this. So in terms of new viruses, how made? Okay, so question mark. And the point is that the virus gets attacks the cell, puts this nucleic acid in the cell. So the nucleic acid of the virus is into the cell that it infects. And then that information is used to tell the cell how to make new viral DNA, or nucleic acid and new viral protein. They get together. By the way, just for interest sake, and you don't need to know anything about this, viruses self-assemble. There aren't any enzymes that say, take this part, put it here to make this. No, they self-assemble. You have millions and millions and millions of pieces floating around, and for various chemical and physical reasons, they get together and form the new virus. They make a lot of mistakes. Who cares? There's so many that work out, And typically in a mammalian cell, you get infected with a virus. One cell gets infected. There might be 50,000 new viruses made inside that cell, and then the cell might die and release 50,000 viruses. Of course, that's not the only cell that got infected. That's a huge amount of viruses when you think about it. If you're talking about a bacterium who gets infected by a virus, they might typically give off 100 or 150 new viruses. But bacteria are small. Our cells are huge, so viruses are incredibly small. You can't even see viruses unless you have an electron microscope. Anyway, so the nucleic acid of the virus is in the cell, and this information tells the cell how to make. I'll just say tells the cell to make new viral nucleic acid. And new viral proteins. And then these get together to make new viruses. And as I said, without going into all the details, I don't think we need to for our purposes, but new viruses are made. How do they get out? Well, in many cases, the cell dies, and then they're just released. In other cases, the cell buds out the way I showed you and takes a little piece of membrane, and then it's an envelope virus. Now, the only last little bit I want to tell you about is just for interest's sake, the flu. Just because it comes around every year, and I highly recommend getting the flu shots. I had some of the really nasty flus that have gone around. I had the Asian flu, I had the Hong Kong flu, I don't know, they don't even name most of them, but whatever. And what happened, for various reasons, the flu virus, there can be a lot of mutations, so you get new varieties of it, and also you can get a mixing of human flu viruses with animal viruses, animal flu viruses, pig, bird, swine flu, and
H1N1,
avian flu, and all that bird flu, and then you can get a mixture of these things, so we can get new varieties of flu, so we can always get new ones, and now you read about it, and H1N1 and all this, just for your interest, you don't need to know this. I'm just going to tell you this, but on this envelope, how did you get the envelope? By budding out of itself, a little bit of membrane. But I did say there's some viral proteins in it. So I'm going to put a little line here, and I'm going to say for viral proteins. Now, this is what I'm going to do is talk about flu virus. And flu actually happens to be helical, not icosahedral, but that's okay, no big deal for our purposes. But in the flu virus, this is just something for interest, you don't need to know this. So the flu virus, which happens to be a negative RNA strand, you don't need to know that, is enveloped. And two of the proteins in the envelope are called H and N. Now, of course, that's just short for the entire word, which we don't need to write down. H is hyaluronidase, and N is neuraminidase. Doesn't mean a thing to any of you, I'm sure.
Thanks.
But here's the deal. It's these are, when I put these little lines here, this could be like an H, a certain H, H1, this could be, this could be N2 right here, another H1, and here's an N2 or something like that. So, but there are a whole bunch of varieties of H, hyaluronidase, a whole bunch of varieties of these N proteins, and it's these that allow this hook onto your cell and allow it to get into your cells and cause infection. But it's also these that your immune system reacts to, or reacts against. So that if you've had the same variety like last year, whatever the variety is coming around this year, if you had that same variety, you won't get it again. If it's a different variety, you will. But there's a whole bunch of different H proteins, there's a whole bunch of different N proteins. So one kind of flu virus, they call it H1N1. But then there's also H1N2, H1N3, H2N2, H2N3, et cetera, et cetera, et cetera. So that, I'll just say et cetera, because there are all kinds of different ones. And so that's the way they classify these viruses. This is flu, the flu virus. And so when they make the flu vaccine each year, what they do is they start monitoring. As soon as flu starts showing up around the country, they start monitoring it, and they go, okay, what varieties are they? And they check out the varieties with respect to the H and N proteins. And they say, okay, hey, look, we're getting some H1N1, we're getting some H1N3, hey, we're getting some H5N2. So they quickly get those viruses and put them in a form that they can put into the flu vaccine, and they make a whole bunch of that vaccine. And then if you go to get a flu shot, you'll get whatever varieties they think are going to be showing up. It's a guess. It's a guess by monitoring, but so they'll be right in some of the cases. But you can get the shot, and a different variety might show up that wasn't in that flu vaccine. They didn't know. I mean, how do you know for sure? But one came from another country that you weren't expecting, So you can still get the flu, but it would be a different variety from what's in the flu vaccine. So I've heard people say, I got the flu vaccine, didn't do a bit of good. It did some good against the ones that they had in there. They don't know exactly what's going to be in there. They can't put every single variety in the flu vaccine. My God. You can give a gallon of vaccine. No, I'm kidding. It wouldn't be that much. Yeah, you had a question. Go ahead.
I think it was just like, are those variations any more powerful? Because there was a big scare. They all do the same thing. They all generally kill old people or young people.
Yeah, and the flu
they don't affect the major population.
Well, and by the way, the word epidemic refers to the fact that a whole bunch of people get higher than normal level. That's when they call it an epidemic. If it's going all around the world, they call it a pandemic.
Mm-hmm.
And the first one that they really found out about in serious terms was right after the First World War, 1918, the Spanish flu. And it killed like 18 million people around the world. It spread around the world. It took like 2.5 years to spread around the world, but it spread around the world. How long do you think it takes for these things to spread around the world now? Well, probably several weeks. Probably.
People don't wash their hands anymore.
Oh, we have airplanes.
But
it's got little to do with washing your hands.
You get sick. You don't know that you're sick yet, but you may be. The weird thing is that most of the time when you get one of these viruses, you are actually spreading the virus before you know you're sick. And then you start feeling sick, and then
breathing spreads viruses.
Spreading to anyone maybe. But in the meantime, you've already infected everyone. You don't know this. It's not your fault, But anyway, so now with rapid transportation, you can get on the plane, travel to another country and born. So these things go around the world now in just a matter of weeks, just a few weeks versus a few years it used to take. But the one in 1918 killed like 18 million people. It was serious. They don't know why it was so virulent. And they actually wanted to find that virus. I was going to say H1N1, H1N2, etc. Just all these different ones. H2N1. Just all these varieties. And I forget which one the 1918 flu was. It was H5N5. I forget right now. Maybe I forget which one it was. But they actually wanted to try to find it. Because nowadays, when there are all these diseases, they will take tissue samples, blood samples from people, especially if people are dying. Then they'll take tissue samples and blood samples and all that, freeze them, and then they can study them. But they didn't have any back in 1918. They didn't do this, so they actually went in various places around the world to try to find where they might find bodies that still had it. So one of the places was Alaska, and they could get some of the bodies, in some of the villages. They would just dig down a little bit and put it, bury them. But it's constant permafrost. It's constantly frozen. So they actually went and tried to find some of the bodies and dig up, and take some tissue samples and see if they could reconstruct what that virus is, so they could be ready in case something like that or something similar to it, came out. And I think they were finding, and they might have gone to Siberia too, again, because of the, cold, so the bodies didn't deteriorate. They could get some tissue samples. I think they finally found out which one it was. I can't remember the details, but it's just kind of weird. The only other thing I want to mention, then we'll go on to bacteria, and that is that one very terrible viral disease has been wiped off the surface of the earth, and that's smallpox. Okay, smallpox was a good one to be able to get rid of because it only hits humans, doesn't hit any other animal. There's no animal reservoir, and they had a very good vaccine, which actually was started back in the late 1700s by Edward Jenner. Using cowpox. What's the Spanish word for cow? Vaca. Vaccine. That's where the word vaccine comes from. It's actually comes from Latin, but that's where the Spanish word comes from. But that's where the word vaccine comes from, because he used cowpox. It turns out that milkmaids milking cows would often get these little infections on their hands, and they didn't get smallpox. And everyone else in the villages in England got smallpox. And 50% of the people died who got smallpox. So he took these little scrapings from these little milkmaids and put them onto his son to begin with, because his son hadn't had smallpox, and his son didn't get smallpox. He tried on others and it kept working, and so they do get a little cow, but you don't get sick. You just get a little, little swelling,
the same type
stuff
of virus
happening right in your hand. But now, when I was a kid, which we got in our arm, now they don't even give the vaccine anymore. You don't need to. But anyway, they finally went around the world, getting rid of all the smallpox in the world. The World Health Organization did it. Took 8 years or something like that. And most microbiology books will have the picture of the last person with smallpox. Somehow, I'm not sure that that's something I'd want to be known for. But anyway, here's the last person. It was in Ethiopia. The two big reservoirs of smallpox in those days were Ethiopia and Bangladesh. So they went to those areas, and they were able. Anyone that got sick, they quickly went and vaccinated their neighbors. They couldn't vaccinate everyone. It's too big a job to vaccinate everyone in the world. But anyway, I said that they've wiped that disease off the face of the earth. That's not totally accurate.
Nope.
The United States and Russia both have vials of smallpox frozen away in some labs. That was back in the days of the Soviet Union, and that was a way to say, okay, you don't try to, do biological warfare and give us smallpoxes. We'll give it to you too. So anyway, so that is still the next the one they're trying to get rid of, and they hope to get rid of this in the next, I don't know how many years, is polio, another viral disease. With polio, the only animals that get it are humans and the great apes. So gorillas and chimps can also get it, but we're the only ones that get smallpox, for example. And all these other viral diseases. Ebola is another horrible one. It's a nasty one. It kills most of the people who get it. They can't find the animal that is the reservoir that has it, but it can cross species line and infect humans, and it's deadly. It's horrible. So these things are. What's scary to people in the public health fields is that as humans spread out into new environments, especially some of these big forested regions, some of the rainforests and whatever, suddenly humans are being exposed to all kinds of situations where they weren't there before, and they may suddenly change the habitat and the ecology and get exposed to viruses that we haven't been exposed to before, and we don't have any natural immunity to, and so it can be pretty bad. So that's always a big scare. But anyway, enough on viruses. Fascinating little thing. Absolutely fascinating critters. Critters. I don't know if I call them critters, but whatever. And by the way, every organism, every cellular organism on Earth that they've studied to this degree, they found that there's some virus that infects them. Now, they haven't studied all organisms, but the ones they've studied the most are animals, plants, and animals, including insects, and then bacteria. But they found viruses that attack protozoa and all kinds of other things. So, and there's a reasonable idea that maybe any organism, there's maybe some virus out there that might be able to attack it. Where do viruses come from? Nobody knows. But there are these things, these little elements, and we have some in our genome, not that we want them, and they're called transposable genes. Transposable genes are sometimes called jumping genes, if you've ever heard the term jumping genes. And these are little bits, I'll get you a question, one second. These are little bits of DNA, and the way they want to be replicated. Okay? Now, here's the thing. If you look at DNA, you could say that what is DNA's what's its function in life? To be replicated.
Yes.
Okay? And what is the human body? The human body is a repository of all kinds of DNA, and it's such that if it can find a mate and it forms new organisms, new offspring, then you're replicating all that DNA. Maybe only half of your DNA is being replicated, but still, you're replicating it, okay? So you can look upon if you're looking at everything from the point of view of DNA, you can say the body is just a way to get the DNA to be replicated. I'd like to look at the human body a little bit, a little bit better than that. But this is a way of looking at it from the point of view of DNA. So anyway, so there are these elements in our genes that only seem to be there to be replicated, and being part of our genes or being part of our chromosomes allows them to be replicated. But every so often they call jumping genes because every so often they'll come out of. So here's a gene. There's a little point right there. They'll come out of that gene. They'll move around, maybe get into a new gene. They could interrupt the gene. They could actually cause a problem. But anyway, they don't care. They just want to be replicated. So what they think is that maybe those transposable, they're actually called transposons, is what they're called, or jumping genes. Maybe those were one time. Maybe that's the origin of viruses. Just some DNA elements, or maybe RNA elements. They quote, want to be, if I can use the word want to be duplicated, and they somehow picked up some genes that allow them to, make that protein coat and whatever. There's all kinds of experimentation going on, so it's not unlikely these things happen. You had a question?
Yeah, you said that viruses affect all cellular life or not.
Yeah. So there are viruses that infect fungi. Yes. There are viruses that infect all. They found whatever when they've looked hard enough, they found. They just haven't looked that much. But
people will go looking for
okay. Well, I'll tell you about
for viruses, but yeah, they have.
Next lecture about virus. But there are let's just say that any of those organisms that they've looked at sufficiently, they found some viruses that can attack them. And so we just say, hey, maybe it's possible that you pick the organism, there's some virus somewhere that can attack it.
Like aspergillium that's made from milk. Like there's viruses that you'll find in there because of different.
But no one knows that that's true. That there's some. It's not an unreasonable idea considering that every time they look, they found something. Let's go because we don't have too much time, and I want to say a little bit about bacteria here. Bacteria. What do we know about bacteria? What kind of cells are they?
Prokaryotic.
Yeah, prokaryotic. They're not very big, are they? Now here's the thing.
They fit in your pocket.
They're very tiny compared to our cells. In my lab last week, we we collected bacteria from like our bodies and stuff like that and grew them onto petri dishes. And like today's lab, we were looking at them and they go, yeah, gross, look, this is off my belly button. Oh God, this is from my cell phone. Look at all these bacteria. There are bacteria everywhere. I mean, that's normal. The nice thing is that the majority of those bacteria don't hurt you. There are some that can cause some nasty infections. Those are bad guys. We definitely don't like those guys.
And then if you make
But most of the bacteria are normal. And as a matter of fact, not only are they normal and just fine, they're necessary for life on Earth. And I've mentioned this many times to you. I told you my little mantra when I teach bacteriology. What was it? Bacteria rule. And they're absolutely critical for all the various life cycles. I mean, cycles.
Well, they can hold
phosphorus
atoms.
cycle, sulfur cycle, carbon cycle, and you name it, nitrogen cycle. They're absolutely critical for all those things. But we tend to think of bacteria as germs that cause disease. Well, some do. But the big thing about them is this diverse metabolism. Now, here's the deal. If I told you to go to the zoo and you point out an elephant and maybe a snake and maybe a rhinoceros and whatever, seal or something like that, you can do it. You can pick them out. They're all pretty different looking, aren't they? But if you check out the metabolism of each of those animals, it's all pretty much the same. Good old Krebs cycle and everything else. Pretty much similar metabolism. So eukaryotic cells are vastly, let's say multicellular organisms, vastly different with respect to their shape, their structure and all that, but very similar metabolically. But if I go to bacteria, they're very diverse metabolically, but they all look alike. Well, not quite. That's an exaggeration by all looking alike. But most of them only come in like 3 shapes. So bacteria, mainly only 3 main shapes. So they look alike. That's what I mean by look alike. They're really pretty much similar. But what I was saying about diverse metabolic. So, so if you, like in a microbiology class, you isolate a bacterium, you look at it, can you tell exactly what it is? No. But you can say something about its structure, then you can do tests to see, hey, what kinds of things does it use for food sources? What kinds of products does it give off as waste products? Because that's vastly different. We talked about 2 different kinds of fermentation. There are dozens and dozens of different kinds of fermentations depending upon the bacterium. So I mean, they're really incredibly diverse. Just about anything you can think of on this planet, there's some bacterium probably that can use it for an energy source. I mean, that's just how diverse they are. What about us? It's not true for us. We can use a lot of things, but there's so many things we can't use. There are some bacteria that can actually eat oil. In fact, they use them in oil spills now.
Yeah, I heard about those, but what are they? Those microorganisms that eat oil?
Yeah, I don't know what to call them, but yeah.
Are they real?
Oh yeah. No, they're real. Yeah. But 3 main shapes: rods, spheres, and a helix or well, it's a spiral. That's what I wanted to say, spirals. And just for your interest, we don't need to get into this, but the rods are just they're longer than they are short. Spheres would look like that, and spirals. Like that. The word for a sphere is coccus, and so you've heard of streptococcus, staphylococcus, strep throat, caused by streptococcus, and the coccus word means sphere. It actually means grape, but because it looks like a grape in the microscope, a little round grape. But anyway, but those are the 3 main shapes, and then as I said, diverse metabolically. I want to give you so let me say something about. Most bacteria have a cell wall. There are a few that don't, but most bacteria have a cell wall. And of course that gives it strength and all that. against the protection against the environment. But just for your interest, again, nothing you need to know about. You don't need to know any of the details. But that they have a way of testing for the cell wall, and the guy who started this way of testing it, his last name was Graham. Actually, he was Danish. It's more like Graham. I can't pronounce it. I don't know how to pronounce Danish, but we just say Graham to make it easy. And so we talk about gram-positive and gram-negative cell walls. All that says is, hey, there are two different kinds of cell walls. Nothing you need to worry about unless you take microbiology. So they're called gram-positive and gram-negative. And all the reason why this is important, that's what they're called. And as I say, you don't need to, I'm not going to ask you about gram-negative and gram-positive. But the reason this is important is that depending upon the kind of cell wall, different kinds of antibiotics are more effective or less effective. So it's very important if you have an infection to find out what kind of organism you have. You know, you're being infected by, if it's a bacterium. By the way, if it's a virus, there isn't much you can do. Antibiotics don't work against viruses. There are a few antiviral drugs that you can use, but they don't have any just like antibiotics that are so useful against bacteria. When they first discovered antibiotics, penicillin being the first one, they thought, oh my God, we're going to wipe out all the diseases, all the bacterial diseases. And certain antibiotics are particularly good against some of the gram-positives. Certain antibiotics are particularly good against some of the gram-negatives. But it didn't wipe out all the bad guys.
Can't find spirochetes.
Because they reproduce so fast, mutations occur, and what's happened? Resistance. Resistant organisms. And it's a perfect example of Darwinian natural selection.
Yeah.
You have a bunch of bacteria. Here's a big glob of bacteria. You start hitting with a certain antibiotic, and most of them are susceptible to it, so they die off. But a few of those organisms might have a mutation that makes them a little bit resistant to it.
And
so
those reproduce.
And reproduce. And so suddenly you have a whole population of, resistant bacteria. So anyway, it didn't happen that we got rid of all of them, unfortunately, but still, there's still some good antibiotics used against all kinds of things. And that was, by the way, it was when antibiotics first came about that doctors became miracle workers. Because in the old days, doctors knew all about diseases. In fact, this happened with Pasteur. He was an incredibly good scientist. He was a chemist, but then he started doing all the work on bacteria and stuff, viruses. And his favorite daughter got typhoid. I think it was typhoid. Anyway, some disease. And he said, the doctor says, well, here's what's going to happen today. This is going to happen tonight. She's going to have this high fever. Tomorrow she'll die.
Typhoid fever, yeah.
God, he says, can't you do something? The doctor said no. And that's suddenly when Pasteur said he was going to try to investigate this. He didn't like that answer. No, there's nothing you can do. His daughter did die, unfortunately. But he went on to study these things. He developed all kinds of vaccines to prevent, and in some cases treat. And they still use some of them, like for rabies. Rabies, they still use the Pasteur treatment a lot. There's some other similar treatments, but Pasteur treatment is still used. And so it's kind of amazing thing. But the point is that when they discovered the antibiotics, suddenly you could take someone who was going to die of some horrible infection, give a heavy dose of antibiotics, and they'll suddenly get over it. And that's like a miracle, okay? And it was a miracle in that sense, As I said, now, of course, there are all these resistant strains, and that's worrisome, very worrisome to the public health field. Question is, yeah.
How do antiviral drugs like Tamiflu and stuff like that work?
Yeah, no, I have to make sure. I remember taking Tamiflu once. I think Tamiflu, and I may be not saying it correctly about Tamiflu, but some of the antiviral does. And Tamiflu may be one of them. It prevents the virus from attaching to the receptors. It keeps them from getting into the cell. And that's usually the way they work in some way to prevent that. Because once the virus gets inside, or at least gets this nucleic acid inside the cell, it's pretty hard to do much about it. But so most of them do that, or they work on sometimes certain certain viruses use certain specific enzymes, and if they can find some drugs that will knock out those enzymes, then it might not be able to reproduce in the same way. So there's a couple of different ways. You can look up Tamiflu now that you know a little bit about it, and if it gives a little bit about the mechanism, you'll probably be able to understand it, you know. But anyway, all right, but back to the bacteria. So the one thing I want to do for interest's sake is this. First of all, they found bacteria just anywhere on Earth. You name the environment, there's some bacterium that is living there. Well, I have to be careful about the extreme conditions because a lot of those extremophiles now are in a separate category of archaea. And we'll talk about that next time. But still, there are bacteria just about any and all kinds of habitats, or at least they're prokaryotic cells in all kinds of habitats. But the one thing I want to talk about that I think is kind of interesting is oxygen. We take oxygen for granted because we need it. We actually can't survive without it.
Yeah.
In fact, your poor little brain, no, your poor big brain, 6 minutes without oxygen and those neurons die off. So that's why, you know, we keep breathing. don't hold your breath too long. But let's just talk. I want to talk about O2, use of O2 as a comparison. This is a comparison, you know, by different organisms. Because you just automatically think probably, oh, everyone uses oxygen. I mean, we do. Eukaryotic cells do, right? So let's take what we call aerobes versus anaerobes. Oh dear, anaerobes. You know, when Pasteur and some others were first studying bacteria, they didn't even know bacteria existed before 1800 and something, whatever the year was. But when they first started studying it, they were just collecting bacteria, like they take bacteria from food, feces, and grow it. There's all kinds of bacteria there. Then someone thought, what if some of these bacteria don't use oxygen? They'd be anaerobic. Then we have to culture them in a different way. It turns out the majority of the bacteria in your gut are anaerobic. But let's go aerobes. So we have aerobes. Let's see. Let's do a little more room here. So aerobes mean they need oxygen, right? Need oxygen. Anaerobes don't use oxygen. So we're what we call obligate anaerobes. That's you know, eukaryotic cells. We have to have.
No, we're obligate anaerobes.
I'm sorry, I didn't mean to say anaerobes. Thank you. God, I'm not paying attention. Thank you. Megan. Obligate aerobes. We have to have O2. So that's eukaryotic cells in general. Okay. But there's some that need oxygen, but not the same amount as is in the atmosphere. Those are called microaerophilic. Microaerophiles. Microaerophilic would be the adjective. These use O2, but a lower concentration, as in the atmosphere. Where would you find these guys?
Higher altitudes?
Maybe higher altitude, or
No.
down in the lakes or oceans or something like that. Not too low, but not too high. Too high, you're like the air. Too low, you don't have enough. Somewhere in between. So these are various bacteria. One other ones are called facultative anaerobes. And I could put these under either category, but I'm going to go with this again. Facultative. Now these are actually facultative anaerobes is the way that they call it. Okay, facultative anaerobes, they use O2 if it's available. But ferment if no O2 is available. So they have it both ways. They can respire, use cellular respiration, or they can ferment. Actually, remember our muscles? Remember we talked about that with our muscles?
Yeah.
Our muscles can ferment briefly if you're trying to exercise really strenuously, but we have to stop and go back to our normal respiration. But there are various bacteria that can do this. But so this is some eukaryotic cells and some bacteria. By the way, I forgot up here, for the obligate aerobes, eukaryotic cells, and some bacteria. So there are some bacteria that are strict aerobes. Obligate aerobes, strict aerobes, you can use either word, means you have to. So those obligate aerobes means you have to have oxygen. Some bacteria fall in that category. Microaerophiles, you need a lower concentration bacteria, some bacteria. Facultative anaerobes, some eukaryotic cells, some bacteria. Now we go to the next category, which was anaerobes. These guys, as I said, don't use oxygen. So they're the obligate anaerobes. Obligate means they have to have anaerobic conditions. They'll die in the presence of oxygen. Where are they going to be? Well, maybe deep in the ocean, under the soil, whatever. Some place where there isn't. There are a lot of places on Earth without oxygen. Surprisingly, but that is the case.
So they could be on the moon?
So perhaps we don't know. So far they haven't discovered life on Mars.
Oh yeah, I know. But I was just saying.
So again, some bacteria. Some bacteria do this.
Spent a lot of time up there since
forms spores, but we're not going to talk about that. And then there's the aerotolerance. Now you don't really need to know all the details here, except I'll tell you what I want you to realize. Aerotolerant anaerobes. These don't use O2, but O2 doesn't hurt them. So this is sort of weird, see? And again, these some bacteria are these.
Aerotolerant, huh?
Aerotolerant. So they don't use the oxygen, but it doesn't bother them, so they live freely in the place where there is oxygen. They just aren't using any of it. So, but notice about bacteria. Every category, there's some bacteria in that category. Where are we? We're really in the obligate aerobes, although for a short amount of time our muscles can fit into the facultative anaerobes, right? For a short amount of time for strenuous exercise. But bacteria fit into every category. That's what I mean. They're very diverse. That's just with respect to O2. I mentioned, all kinds of fermentation pathways. All kinds of things can be made by bacteria. In fact, that's the way they make a lot of products. At least they used to. Some they make, themselves, and chemists do it. But a lot of things they would make by just growing bacteria, certain kinds of bacteria in big vats, and then collecting what they give off. Swiss cheese. Swiss cheese has those holes in it, right?
Mm-hmm.
Good Swiss cheese, those are natural. Cheap Swiss cheese, they manufacture the holes, but good Swiss cheese is natural, and what happens is a certain kind of bacterium they use to make the Swiss cheese, called Propionibacterium, and it gives off some propionic acid, which gives the flavor of the Swiss cheese, and it gives off CO2, which causes these
bubbles.
spaces
Yeah.
come out, when the cheese is hard, the little bubbles. One last little bit. Oh God, you guys are putting your books when I got 3 minutes. Oh, 2 minutes. Golly.
It's because they know I'll stay.
Bacteria and humans. One last little bit about this, just real quickly. As I said, the vast majority of bacteria are not bad guys in terms of causing infections. They're normal kinds of things in the environment, and many of them are absolutely critical to maintain life on Earth as we know it. But in terms of your body, we have all kinds of resident bacteria on our skin, all kinds of resident bacteria in our guts. Now, here's the thing. When I'm talking about bacteria in our guts, that's not in our body. Remember I talked way back when about how there's an open tube running from here all the way to the other end, right? So the bacteria in your gut are outside your body topologically. They're just in a tube surrounded by your body. If those bacteria cross the cell membrane, then they're in your body and they can cause an infection. But those guys in your guts, for example, those are good guys most of the time. Now they're said to be opportunists. Meaning if you give them an opportunity to cause an infection, they might. In other words, if they had a cut or something, they got inside, then they can cause an infection. That's not good. But in terms of just living where they're living, they're actually good guys because most of the time they're keeping bad guys from getting a chance to get a hold in there. In other words, they're filling up all the little niches. And so you eat some bad guy, it's not as easy for the bad guy to suddenly say, hey, I want to live here and cause the problem. It can happen, and we've all gotten sick. We've all gotten some nasty intestinal, bacterial infection or something. But in general, those things are good guys. A lot of times, if you take some serious antibiotic therapy for whatever, you have some diarrhea and problems like that. Of course, if you're taking some serious antibiotic therapy for some horrible infection, you might be killing off some of your normal intestinal flora. So after that, they usually say you want to replace that with more good guys. You can take yogurt, you can do what's kefir, what are some of those other things that you can get?
Lactose intolerant people do.
But anyway, but yogurt's a good one to take, was it? Yeah, that probably has some good guys in it.
That fermented seed stuff.
Yeah, fermented the Korean fermented tea kind of.
It's weird.
Anyway, but those are they often say take the good guys. But what's the other one they say to take? But these guys are they get in your gut and they again kind of repopulate. So and the essentially what we're talking about is probiotic. Yeah, and well now that's a.
Well, no, it's the one from yogurt that you're talking about.
Yeah, but anyway, so those things are actually healthy for you.
Healthy bacteria.
All right, you guys.
All right.
See you.
All right. We've only got 4 of these left. For those of you that weren't here today, we had a quiz. If you missed it, you get an F. Bye.
You're fine.